Hello and hello everyone, welcome to class 11 biology. In our previous video we were discussing about metabolism. I told you there are different types of chemical reactions going on within the body of a living organism. Let it be a single cell bacteria or a multicellular organism like us, right? In all the bodies of the living organisms, different types of chemical reactions are going on, and all those chemical reactions are necessary to keep the living organism alive. Right, to keep that organism alive, those chemical reactions are necessary, right? And all those chemical reactions, they are called as metabolism, and therefore metabolism is synonymous with living state, right? So that's what we discussed, and at the end I told you that all these chemical reactions are catalyzed reactions. That means the chemical reactions, they are hastened by the presence of a catalyst, and those catalysts within the body of a living organism are enzymes. So in this particular video, we will be discussing about enzymes. Now, how do we define enzymes? Enzymes, the simplest definition is enzymes are biocatalysts. The catalysts which are found inside the living organisms, biocatalysts. So enzymes are biocatalysts. Enzymes are biocatalysts. The enzymes proteins. Almost all enzymes are proteins except ribozymes. Ribozymes, they are not made up of proteins. Please find more information regarding ribozymes. Ribozyme kalatis nezu mangsia. Ribozyme di kharigi dubsha yoro to sia kiranzo. So ribozyme mado shimba enzyme mangche kharigi dubsha yoro lare proteins ki dubsha yoro. Ta kisi enzymes sama proteins ki dubsha yoro yimba ina. Ani kola primary, secondary, tertiary structure yoro. That enzyme ki tertiary structure di. Enzyme ki tertiary structure dila. Three-dimensional structure dila. And they have got many pockets or crevices. Crevices da way mana ta bench the tertiary structure of an enzyme imba chasha. And ko gala khari three-dimensional structure le du zambala. Tertiary structure le du zambala. Ko gala and crevices la when ma thimjo rom the valley stone la war. And pockets la way mana ni saja shongsa and la war tongba and la war. So tertiary structure of enzyme has got many crevices or pockets, right? And one such pocket on the enzyme galo yo the pocket dinde chikwa tilang azu khar lagre la way mana active site of the enzyme ki active site measure. The active site depending on active site is very important because active site is the site into which the substrate can fit. Active site na lola substrate takta fit chiwar. Ta substrate lana khala war. Substrates, they are nothing but the reactants of the reaction. Reaction chik na lola khar gugore. Reactants gugore. Reactants dige react chewe kengi ani product form chigro. Inashi the biological reaction now reactants is substrate enzyme ki substrate. So substrate dige sama enzyme gi pocket fit chigor pocket fit chesa dilanga zu khala ore active site la ore this can theba yagu nyama do can remove dil sin digidu remove din nanglo la seu ji do di kharre enzyme gi tertiary structure na lo de la sa tongba de do de saja tongba sto dilanga zu khala ore active site la ore and active site din nanglo la khar takta fit chigido de si the jangu di kharre substrate re reactant ko del takta fit chigor lock and key nangshi dimi da konja nangshi takta de fit chig di fit chesa we jela ani enzyme di ko gala jar ani ko khar chigore reaction di catalyze chigor reaction di gyor tangor seu chungso so enzyme gala active site jores active site di kharre ye to kharre enzyme the protein into zambalo ko tertiary structure nanglo la three-dimensional structure de nanglo la saja tongba and mangbo yore saja tongba de zu nanglo ne chigala khar chigore substrate takba fit chigor di fit chesa de langa zu khala ore active site adi kana stemba yagu nyama to ta enzymes the proteins ki dubsha yore to ta kisi enzymes the proteins ki dubsha yo yimba na proteins mang che khare temperature sensitive yor temperature tho shu cha we yimba na protein the denature cha ro denature cha wala na proteins get damaged. Proteins, they get damaged at high temperatures, which is quite different from inorganic catalysts. Catalysts shemba the mandawegi, enzymes which are biocatalysts, they are sensitive to high temperatures because enzymes, they are made up of proteins. Proteins, they get damaged in high temperature. Temperature so you zambala khachore enzymes the thola do and they are chetu go so that is here. However, enzymes of thermophiles, they retain function at high temperature up to 80 to 90 degrees Celsius. Enzyme gashe kharche gore 80 to 90 degrees Celsius le lega yagu chetu gore. Ine 80 to 90 degrees Celsius temperature de thola lega chetunge ki enzyme dinga zo kharza ne ragare la wo yimena thermophiles san ragar. So thermophiles la wo yimena those organisms right, which can withstand high temperature or which lives in a high temperature environment. The bacteria which are found in hot springs. Chuzen nanglo la ne thunge gi bacteria and yorwa. O de zola ngazu khar la chore thermophiles la chore. Thermophiles, they are organisms which loves temperature. Philic la women ngazu khalo thermophile la women temperature loving organisms. Okay, so the enzymes of temperature loving organisms like the bacteria which are found in hot spring, they have got enzymes, right, which can retain their function. Konsu khache tu gore temperature chau yunege lega chetu gore tando lega temperature chau yuzambala lega yaga chetu gore. So these are some of the characteristics of enzyme. Now we will discuss further. Okay, the enzymes ki kharchi gore ngazu subu nanglo yo chemical reaction dige jor tang gore slarwa ta desia ngazu reaction chi latta wi ba na reaction nanglo reactants the products yore la ba kera zunyama jang yu gil chunchu gap you must have studied in your lower classes. In a chemical reaction there are reactants they react with each other to produce product form che che e chi baji gen delare a plus b kharcha so c chag do a the b ni di kharcha re substrate char c di kharcha re product char or a jor da ko kharcha re b plus c char a di kharcha re substrate char re b the c ni di khare product re tai nyama ngazu gi metabolism ga jang du zambala anabolic reaction da catabolic reaction an jang ba di kharcha re anabolic building up process a the b ni dia jar ni ani c cha so che cha so the anabolic reaction ni ma cha cha di khare catabolic reaction di che di chung ni ma cha so so ine da del zo khar go che go wala la wi ba na a the b hydroxide yu zambal ko kharcha re substrate enzyme gi substrate cha du re A che ku de chu yo bi mana ro B the C produce chi yo bi mana ya A di khar cha re substrate cha do se chung so now the reaction ro substrate the product lan to. Now remember the amount of product formed per unit time is called the rate of reaction. That reaction is do ya jumar A plus B C chayegi speed yore A ne B the C chayegi call reaction speed joro this speed dilanga zo khale rate of reaction le or the rate of reaction or the speed of reaction. Speed of reaction ding also khandes calculate chigre la win na ngazu gi product gi concentration the measure chain the amount of product produced in a unit time tizu tis na la product khazu produce chido ding ngazu gi che ya to win na ani ngazu reaction gi rate joro ding ngazu che ya to so the chemical formula the position deal the ngazu mathematical formula the rate of the reaction is equal to delta product divided by delta time. But delta product now we increase in the product, right, divided by time taken for the product to be produced. Rate is equal to delta P by delta T. The rate dila effective thunge roa. So effective thunge chemical reaction mangchela roa. Rate of reaction jomarwe. Dila effective thunge so mangche khare la na temperature chadur. Temperature is one of the factors which affects the rate of reaction. Rate of reaction temperature ya thor du zambal khachi gore rate of reaction jor joya se chung so chemical reaction mang channel la worda when the temperature increases the rate of reaction also increases khachin re because temperature they provide energy for the reaction to take place se chung so ani kya lo te bya lo yo bi mena dinal khar chi shala bi mena degree centigrade chu phar du zambal la ro pejin reaction rate double chagre la yo t-shirt kya ya go chen go ne lo da ro diche ani the main begi temperature main begi ya khare they are catalysts or enzymes. Catalysts or enzymes they can vastly affect the rate of reaction. The rate of reaction the dab gibat kore ya par thungi gi nuba de sule yore law imbana the catalysts enzymes tigeliyor. Se chung so catalysts in case of inorganic reactions, whereas in case of organic reactions, in case of metabolic reactions we have got enzymes as biocatalysts. And enzymes tige gi kharche tu gore reaction gi rate joro the dab gi ya par tu gore. Let us look at the power of enzymes and let us take an example. Example chikhiche an enzyme gi nuba ditta da. Reaction chikte da delasia carbon dioxide plus water. Carbon dioxide plus water kharla change do carbonic acid la change do. The product re dini khare reactants re dini substrates re. Enzyme may vegi kabla. When there is no enzyme present, when there is no catalyst may vegi kabla roa. In the absence of any enzyme, the reaction choroa carbonic acid form chayegi reaction chomare kope thevoyor it is very slow, right? Only 200 molecules of carbonic acid is formed per hour. Chujeching nanglo la roa carbonic acid nige modu form chedu womar. Di khaju mangbu yobi mene carbon dioxide the water khaju mangbu yobi in by ne reaction take place chela thuju shu gorgor chuju chig nanglo la carbonic acid niga madu form chetu woma tang azu reaction di nanglo la enzyme jiglu bi in by na carbonic anhydrase la ki enzyme di lu bi in by na enzyme di khare biocatalyst ra ko hachu gore reaction di speed jo mare gyor tangro khaju gyor tang du chance de sida in the presence of enzyme called carbonic anhydrase the reaction rate is increased by 10 million times. Dab saya chu thamba ki bat koro gyor tang gorda. Sa dab chik dab ni gaj keja marda. Dab saya chu thamba. Condensation de sida 600,000 carbonic acid per second. Yela per hour nala reda. Chuju chik nalo la niga madu form chetu wo mar enzyme mewe kabla. Enzyme duslu wada khachi gore second chik nalo la 600,000 carbonic acid produce chetu wo. That is the power of enzymes. Enzymes ki power thagarane ha go do ni enzymes khagums ki khechum bui mechanism go chu do so now the inde yimba ina ro let's define metabolic pathways the metabolic pathway thing also can redefine cho thing the metabolic pathway jangsar da when we discussed about metabolism we discussed about metabolic pathways lana khari imbe ro metabolic reaction mangbu chi kharchere interconnected rail line ba so they can form a linear pathway or a circular pathway also jang ba thing de tang azu din nanglo la enzyme ki role ta yamarta tang azu enzyme la kharchi gi mechanism seu chungso di into zambala kisi enzyme ki role di metabolic reaction na kana go ya chhe yo bi ba na ta metabolic pathway la ding azu yangya jikta go a multi-step chemical reaction where each of the steps are catalyzed by the same enzyme complex or different enzymes is called as a metabolic pathway ta metabolic pathway la na khalla gore la bi ba na dinya de she khar jasu metabolic reaction mangbu chik ro pasu interlinked him and chik the chik connected him by na then metabolic pathway la gore line ba ta de la khari yore enzyme la chik di chu yor reaction manda dige sama khari gi catalyze chi gore enzyme gi di chi gore catalyze chi gore ta dige sama khaji they form an enzyme complex right or they form a group of enzymes right and a group of enzymes which catalyze different metabolic reactions right and that type of metabolic reactions they are called as a metabolic pathway for example metabolism of glucose latana digre metabolism of glucose na kharim glucose is broken down into 2 pyruvic acid glucose is broken down into 2 pyruvic acid right tilang azu khala ore metabolism of glucose na tadi dusta we mena simple sado ko do this is not simple right it is very complicated. There are 10 different enzymes involved in the breakdown of glucose to pyruvic acid. That means there are 10 different steps. Glucose pyruvic acid la chaya chedolo step chu thamba yor. Step rare la khare enzyme manda yor. Ta ngazu dinge gi remut gone ta nda ni kranzu mashe anye ngazu jela jangre la imba. Glucose ne pyruvic acid la ma chaya chedola khar gu do de. Step thangbo, step new step sumba lande do. So step chu thamba yor. And step rare la sida kranzu de. Enzyme rare hexokinase it's an enzyme phosphoglucose isomerase phosphofructokinase all these are the names of enzymes involved in the reaction metabolic reaction dine dila change to zama glucose ne glucose 6 phosphate la change che che dula enzyme kharu gu gore hexokinase gu gore reaction ki tor ta che ani glucose 6 phosphate ne fructose 6 phosphate change che che do phosphoglucose isomerase la enzyme de gore din shi din ngombu thijo dige sama kharre enzyme ki ming. So this is one metabolic pathway and metabolic pathway dinalo enzyme chu thamba so these 10 they form an enzyme complex so metabolic pathway chik nalo enzyme complex chik do there are different types of enzymes over here there are different enzymes so chung so the enzyme gi ro biological reaction gi speed the just door the how does enzyme increase the speed of biological reactions. But the goes saying it over the simplest answer was now simple in this complex. The simple answer is enzymes they bring down the activation energy of the reaction, therefore they can speed up the rate of reaction. And simple answer was enzyme gi activation energy laj mama activation energy mama draw by na and reaction gi speed joro the door to the activation energy lana kharle du ta enda. This you can understand from this particular graph. A graph din altani ro kiran zuti len di yapu gozu. Ta this here din nanglo la pink line chik da ro red line chik yor. Yapu sia ro yangya laginda. di nanglo la red line chik do graph di gala khar plot che do red line chik plot che do ini pink line chik plot che do dini ni khare the red line is the one where there is no involvement of enzymes enzyme me wake up la reaction dikhante stugu yeme gaji gi graph re enzyme ye wake up gi ni reaction khante stugu yeme dinga zo kharte ngore pink line ki tengor ta desia di gharre potential energy how much energy is required for the energy to take place for the reaction to take place, right? And this is the progress of the reaction. We are changing substrate right, it is right over here substrate de layer da substrate ki potential energy de layer product ki potential energy dikhayo re manima yor wa del to se uchung so reaction dikhadu du re and the substrate is converted into product reaction chung ro ta desia thangbu ping dilamasia thangbu khaw de mau dilasia enzyme may wege kabla when there is no enzyme involved in this particular reaction of conversion of substrate into product, right? Look at the red line. How much energy input is required? Energy khazulu do they asida substrate nalo energy pe mangulu ra substrate dena energy pe mangulu substrate dena energy mangulu wekengi ani the peak level chila ligi do the peak level dena ngazu khale the transition state level ra substrate energy level de imba the dotted line there do substrate the reaction go su wala in order to substrate to start transforming into product, in order for the substrate to start transforming into product, it has to reach this transition state. Energy lower. Reactant ne product form chewala energy lower. Tadesia energy lower energy lu zaman energy mangnu khazulu de sida substrate ne transition state layer la energy pe mangulu do enzyme medu zambala. And transition state layer da ani lamsang khajire substrate di product la gyur. The energy required by the substrate to reach the transition state is called as the activation energy, and the activation energy without enzyme is very high. The activation energy without enzyme is really very high. Now if we add enzyme to the reaction mixture enzyme lu wi mai na ta desia substrate rangjar da thanda ta khona ngazu kharlu re enzyme lu wa kengi and the substrate the reaction transition state layer la energy shera go marte sida there is not much energy is required tithik the rinze de sida dithik the rinze de sida activation energy required with the presence of enzyme is very low the induzambala any reaction dikhachi do so therefore enzyme they speed up the biological reaction by bringing down the activation energy of the reaction energy of the reaction the mamma rutang wekengi ani ko khachi gore reaction di kyong down to water slash now this is very important the concept the pe khechambur da nenga chambur da kiranzo the remo the yabu chen jang you have to practice drawing this diagram at this particular graph and try to understand what it is trying to tell you is ko ma chyo bi by na ro na zo lotem na yabu chen si ngala thiwa yang ja taw na chi du ro ya ta deshe mu thye inda ro ta din na lo ga zo khar ta la bi by na mode of enzyme action enzyme gi la khandis chi gi me ro digang ta Tadina theory ni share. Theory ni dikhare lawi mana lock and key theory and induced fit theory. These two theory explains the mode of enzyme action. In this particular theory, lock and key theory, the shape of the active site of the enzyme and the shape of the substrate they fit like lock and key. Konja da dimi ni the fit shewa nangshi substrate da ani enzyme galo yu ki active site jora kuni ni khaji gore dimi da gonja na ta fit chigor ro dit nenga chimbu per ta dinna lo deshe dekhare enzyme gi khejo khejimbu shuji deshe gyogo ti khare law imana enzymes they are very specific enzymes dige pe specific yor enzyme changma gi reaction changma lo judo chigmar ro enzyme they are very specific enzyme gi khaji re reactant wo she ani shine ko khaji re odela ani ko judo chigma do ro reaction changma lo ko judo chigma da so enzymes they have got specific substrate because khare substrate gi sobda na active site gi sobda ni dikhaji go ta ta fit che go ro razu gi konji dimi konji khare dimi konja sama na yu yin ban na che tu min do din na shi ji gi yang khare substrate da enzyme gi active site ni sobda ji gi yang yu yin ban ani shine reaction chung go ma do shimba chung go mar the enzyme gi khar chi go re specificity de do and that is very important characteristics of enzyme enzyme gi khejo khechim bu jire ko pe specific yo res ro na desia enzyme gi active site da substrate ni de la fit cheni and kharchi do enzyme substrate complex form chi do tadi nenga chumbur da enzyme substrate complex the form che ti chela khajire enzyme gi rokwa che ni substrate ni di cha shag do ma ni loga su do di kharchare enzyme product complex product form che wa da ni khajire product ni di ko le she ma chilo la so that's how the reaction proceeds reaction ka reactant khari re dingi ro jangu di ro product khare ngombu da seu ni di ro so di para de la enzyme ki kharche so it formed a complex akhoi active site na also le ani reaction di go take place chechu ani product produce ani product ni di pha le so product ni di le wa di kheching bu da ma le yin ba na ya khajo ko next substrate ga la pha ju go wora it has to go and fit on the next substrate it has to act upon the next substrate seu chung so lock and key theory la na seu chung so induce fit la na dina this deshe gyar this ya induce fit la na khala ore lock and key theory da manda khala or la na active site di da ro substrate ni so da chipo jigya yong sig ma res ro dau do yong sig res ta di indu zambala substrate dus jarwa da ani enzyme gi hachi ore substrate dina takta fit che do in the substrate de la jarwe kengi induce cha ani enzyme di takta fit che jo active site dina takta fit che odila khala ore induce fit theory la but you can just remember the lock and key theory kana zo di thimba so na dig digi de she yaga je do enzyme ki specificity gal de she yaga ja do di che ro ta desia di yagu go chyo yimena thanda thanjinye de rangja join da di gal rangja de she gyan da remu do nyam do ro ta de activation energy gal do da the potential energy imba the reaction gi direction imba substrate de do substrate gi energy level de la re do substrate di product maur dir do substrate purple diche product mau di manju ki main goal khar re substrate di product la gyo ta andro kisi enzyme me we in by na substrate di kharche go do substrate di la energy yoy mangu te go do esa yogi bodo ya ke go do enzyme me we kabla no catalyst la we mena enzyme me we kabla la go do idu zambala energy mangu shulu go idu zambala thuju ge mangu shu go and de la le wa da ni shine ko react in la gyo do ro kisi enzyme yoy we in by na if there is an enzyme present enzyme here we by na enzyme di khachire enzyme substrate complex form chir kuni ta fit che weking ti go de del ta fit chi do fit che weking enzyme substrate complex form che enzyme substrate complex ki ani khar la yuru do enzyme product la yuru do seu chung so enzyme substrate complex la energy shira go marte activation energy tho shira go ma transition state lamsan le so di ba so za ani reaction rate di at ko dab ki kyo urtan tu energy khaju gi nyu lu gu yo ne digams ki reaction dikhaji gre kyo go tu yo so energy mangbu shuj lu gu yo bi mana reactant ne product la gyo ta la energy mangbu shuj lu tu yo bi mana ro ani thuju ge mang go ro so enzyme ki kharchi gi do activation energy di ma nyu ru tang che ani reaction kyo ru tang do so dire ta kira zo page number ya da ngab ju ngab ding on your page 157 you will find this particular information given in your textbook, right? This year, the catalytic cycle of an enzyme action. And this described step by step. Now, di malo je ani remu dila si je ni kira go chhe bachita ro thangu dila khar chigirlare substrate the enzyme ni di jargires te jargdo di jarni khari form chigire enzyme substrate complex the form chigir enzyme substrate complex form checha we jela ani enzyme gi diparal yugo chemical bond di ma breakdown cheni enzyme product complex form chigir and di checha we jela enzyme gi khachire product di pha le sa we jela ni ya khachire substrate sawaji jugadu di dela dishe jayorta so kanazu di ma ti ani remu di kanazu ma shunu digidu ro khajin remu di ya go chugdo ina kharchi yu me di ge sama dishe sama dina remu dina yor seo chungso. Now let us discuss about factors affecting enzyme activity. Enzyme activity la affect chingi factor khar khar do. Enzyme lega chidu zambalo ko lega yago chitu matu la dila yuwa tangi gi ani factor khar khar gi effect do ditayen lo. Din alo la factor khar khar yore la in binary enzyme activity affect chingi factor khar khar yore temperature, pH, concentration of the substrate and chemical inhibitors. The one by one ta in binary. If you look at the effect of pH and temperature on enzyme action, let us look at these two graphs over here. This here, pH low yudu zambala, pH pe mau yudu zambala, enzyme ki activity chomare. The sujo side the enzyme activity re wa dikhare pH re. So when the pH is really very low, right? The enzyme activity is also very low. As the pH increases, the enzyme activity increases and it reaches a peak, right? And this particular pH level at which the enzyme activity reaches its peak, it is called as the optimum pH. This is called as the optimum pH. The optimum pH gets a jela. Yeah, pH par dewa imbana ya the enzyme activity ma reduce chadu. Enzyme activity goes down, right? The jela khala ore thanda so khala ore law imbana pH low shu yob imbana acidic shu imba enzyme yabu lega chitu omar. Alkaline shu imba ya pH alkaline shu imba ya enzyme lega yabu chitu omar. Enzyme de la kharu gore they need an optimum pH, the neutral ray la omar da ya. Neutral ray some sigro neutral pH la omar. Kola kharu gore pH the optimum dose gore. And the neutral mare la gumzen khare la imbana. Some acid they work better in an acidic environment. For example, some enzyme they work better in the acidic environment. Wow. So pension enzyme digestive enzyme the digestive enzymes present in your stomach, they function better in acidic environment. So therefore their optimum pH will be somewhat acidic. And those enzymes which function better in basic or alkaline environment, their optimum pH is alkaline. Basic chadur. So din zabadin neutral re lago marda. Ta khala ore optimum pH shoro. Dile mamma thaw imbana enzyme yabu lega chitu mare. Dile bet kora ya tho thaw imbana enzyme yabu lega chitu mar. So that's why this graph is showing you the functioning of enzyme in a different pH scale. Okay. So temperature now when the temperature is really low, when the temperature is very low, the enzyme activity will remain very low. As the temperature increases, as the temperature increases, the enzyme activity also increases, right? And it reaches a peak level, and that is called as the optimum temperature. The temperature at which the enzyme activity attains its peak level, it is called as the optimum temperature. The optimum temperature gets how a J line the enzyme activity down char the graph to smart optimum pH, the optimum temperature. Every enzyme they have got a optimum pH and optimum temperature at which it can function the best. So what do you think the enzymes present in our body enzyme optimum temperature is it the body temperature or is it something else? Optimum temperature of our enzymes. Enzymes optimum temperature I mean research temperature 36.36 the enzyme optimum temperature the effect of pH and temperature on enzyme action. The next effect of substrate concentration on enzyme action. Enzyme action gala substrate magnum gichawa contest to the tire. The graph the graph here the axis over here it shows the concentration of substrate. The x-axis ki concentration of substrate, y-axis ki velocity or the rate of reaction shows you. When the substrate concentration is very low, the reaction speed is also very low. Enzyme ki reaction substrate in a substrate when the substrate concentration increases, right, the velocity or the rate of reaction increases exponentially. At this particular level it will become steady. Steady this steady chasa V max, and the half the velocity half V max ki half the lower. So this V max now the substrate concentration the enzyme activity ni proportional substrate enzyme in a reaction signal in the reaction mixture if you have a reaction mixture enzyme the substrate this chilur enzyme rate of reaction this substrate enzyme gi rate of reaction haji thadu ti sia pargro thadu substrate ma lu bi im baina an haji re enzyme gi rate of reaction di khol li re v max li re di je la khira gi substrate khaju ma lu bi im baine reaction gi speed jo che pargmar seo chungso kharchin re why because enzyme nanglo la limited ro the amount of enzyme within the reaction mixture is limited kira gi substrate lu de du zambala enzyme free in baina enzyme gi active site free in baina ro enzyme gi khachi re koi gi substrate de ma cha ni rate of reaction de apar digir now substrate mangbo shuj lu in baina enzyme gi active site sama khang ru ra juju chigla enzyme sama khaji gi active site de khang ru ra di khang sa we jela delay gyor ta yung maro seo chung se pen cha chana nganzo enzyme in ba cha let's say we are the enzymes okay there are 100 people right 100 people they can do their work with their two hands they lakwa ni gi lega che tu ro ta lakwa re re la chala re re te di gi lega chi gor da ro mi chigla mi re re lakwa ni du zamba la lakwa re re la chala re re te du zamba la lega che tu ta mi changma gi ta azu mi gya thamba yo gyo joro mi gya thamba yo gyo de lakwa changma nang lo la le chala te sa we je la thandwe lega ku ba yin ba na de le la che yung maro tik pik de le sa ro din shi enzyme gi active site di khang sa we je la ani rate of reaction di phara yo mar substrate khazu mo khazu lu bi yin ne seo chu so di lo gor graph di show chi gor ta de la km la sto de ding azu discuss ma chi sha shro di che the next khare effects of chemical inhibitors on enzyme action gala ani chemical inhibitor din zo gi khandes effective yo me tirta ta pena sha sha wi ba na reaction chila sida ro reaction chila the normal reaction dirda enzyme chi yo che digala substrate di jar sa ve jela enzyme substrate complex form che ine enzyme gi product di pha release chi yo ta desia inhibitor ji pheju che wi mana konda inhibitor ji pheju che wi ba na ro inhibitor di kharchi go re active site gala do ni jar go di jar thuye gi nuba di khandes li go la wi mana inhibitor gi sopta di sida inhibitor gi sopta da substrate ni gi sopta di chigba ro jarte ta do min de kunni chigba di ba so za enzyme go the go ro enzyme gi active site gala ko jar thu go right inhibitors are the substances which mimics the shape of substrate right and therefore they can bind with the active site of the enzyme gan jar thu go enzyme gi active site gan jar thu go di jar sa ve jela ko substrate marwa ko reaction undergo chi maro chemical bond break indicate che yo maro an ko khachi gire enzyme gala active site de saja de sim digir saja de sim de du zambala substrate li du zambala ani enzyme de lega che tu go mar therefore chemical inhibitors they can inhibit the enzyme action enzyme gi lega di ka tu go so jo se tu mang che dus lega chi go the most of the poisons the toxic chemicals right they go and bind to certain enzymes thereby inhibiting those enzymes and enzymes lega ma che wi ba na ma ju su bu na lo rate of reaction de ma kyo du le ro ani shi do ani go ro so the effect of chemical inhibitors on enzyme action so there are four different factors temperature pH concentration of substrate and chemical inhibitors now some enzymes which are proteins they require something called cofactors non-protein constituents to be active right and the protein portion is called apoenzyme tadesya wanzu gi apoenzyme chi apoenzyme na khare the protein portion of the enzyme is called as the apoenzyme is inactive but the protein portion of the enzyme apoenzyme is inactive ko lega chetu go mar ko reaction catalyze chetu go mar substrate gana act chetu go mar ko ropa chinge non protein constituents go wor dilanga zu kharle gari labi mena cofactor ro non protein portion de go wor cofactor de khar chigire inactive yo apoenzyme joro ko active sugur the khande sugur labi mena the coenzyme will go and bind to the active site ka jar do the jar sa we jelani structure jik form chig minde di shine khare ni ko khar cha do gore active cha do dilanga zu min khar ta gore holoenzyme so we have got apoenzyme which is not inactive we have cofactor which is the activator so the combination of apoenzyme and cofactor it forms the active holoenzyme so the cofactor the jar wekengi ni enzyme the active cha so substrate nyam do bind chin ko reaction catalyze che tu war tadi activate che chungye jo mara dilanga zu khala gore cofactors la war so the cofactor la rig manda sumyor there are three different types of cofactors prosthetic group coenzymes, and metal ions. If you look at them one by one, prosthetic groups they are organic compounds, right, which are tightly bound to the apoenzyme. You see D raw organic compounds himbache and ko pet ko thambu shuj jar digi yobi na apoenzyme gala. Ko khalla ore prosthetic group chart over. Pencha shawi na heme group, which is bound to enzymes like peroxidase and catalase. Heme group la jo hemoglobin na heme la jo iron containing molecule, right, that is called as heme. Heme ki har chetu ore peroxidase na catalase kala thambu shuj jar tu ore di thambu shuj jar tu weking ani peroxidase na catalase ni har chart over holo enzyme active enzyme chart over. Then there are coenzymes. Now coenzymes like prosthetic groups, they are also organic compounds. Organic compounds with the carbon group shayore. But the bonding of apoenzyme is transient. Coenzyme the apoenzyme gala thambu shu jargo mar. Right? Unlike prosthetic groups, coenzymes they do not bind, right, to the apoenzyme very tightly. Their bonding is transient. Transient and this string jar ani ako hot war. Tinte into zambala ani coenzyme la di khaji gore enzyme manda gala ko activate enzyme manda activate chetu war. Coenzyme chigi enzyme manda activate chetu ya yubayor. So chungsa penachasha wi bana coenzyme gi example chi khare vitamins such as niacin or B3. Niacin is vitamin B3. Vitamin B3 di kharcha do coenzyme cha do. So chungsa the third one is metal ions. Right, metal ions can also act as cofactors. Right, they form coordination bond between the side chains of the active site of the enzyme, and they can also form coordination bonds with the substrate. Zinc. Zinc is a metal ion, right? Zinc metal raw. So zinc can act as cofactor for carboxypeptidase. The carboxypeptidase the zinc molecule zinc molecule active site gala carboxypeptidase ki active site gala coordination bond form che and din nyamdo ko kharchi gore substrate nyamdo le ko kharchi gore coordination bond form che to din zambar ko kharchi do active site gala ko so thing do substrate di so this ni ko yani activate che to zinc can activate the carboxypeptidase. It can make it into a holoenzyme. So now the important point is the catalytic activity of the enzyme is lost when the cofactor is removed. You see, enzyme ga cofactor the pi we in by na ro ni enzyme gi catalytic property jo mara catalytic activity jo ro di me cha do so that is about the cofactors come under sumdo prosthetic group, coenzymes, and metal ions. Any cofactors na khari be non-protein constituents which can activate the protein portion of the enzyme, inactive protein portion of the enzyme. Inactive protein portion of the enzyme dilamazu khala over apoenzyme. So you should remember that. Okay. Classification and nomenclature of enzymes. The enzyme la ming khandes tagi imbe and enzyme dige deze manda la yawa khandes chegi imbe. So based upon type of reaction they catalyze, enzymes are divided into 6 large classes. Ko group 2 lama deze la yawa che ti khari gal shisha ni yawa che go la wi imba na khogi reaction rig khande do ji catalyze che tu do digala shishani ani enzyme dige sama deze thul ma yawa chi resler ro tadi thangbo di khare oxidoreductases and dehydrogenases or dehydrogenases ro so as ye gyo mangche namju khare la gore dige sama khare enzyme reda ro a/ to the suffix as ye gyo dige mangche khare biology nanglo la enzyme chadur chuma kana sem na nyama do ro diche oxidoreductases or dehydrogenases, they are the enzymes which catalyze redox reaction. Redox reaction catalyze chingi enzyme dige lamazu kharla gore oxidoreductases or dehydrogenases. For example, the example te do substrate chik reduce cha substrate s dash dikhare do oxidize re do enzyme dige act chesa vejela thanji reduce ingyo di oxidize cha so thanji oxidize ingyo di reduce cha so ti indu zambala dinde catalyze reaction dinde to catalyze che thunge de jula ngazu khar lagile oxidoreductases or dehydrogenases lens se chung so hydrogen ma pi thunge de na oro dine next kare transferases ata di ming kora ya kare do self explanatory do transfer che thunge it transfers a functional group enzymes which can transfer a functional group reaction in which there is a transfer of functional group dinde reaction ji la catalyze che thunge enzyme ji yo bi na de jula ngazu khar lagile transferases lens then cha sha wi na substrate S di gang la group G la di jar de do de la ni substrate S dash gang la group G yo marta after the reaction look over here S gang ne G di pha ko ne G di khar gang jar do S dash gang la jar do so the group has been transferred the functional group has been transferred to the S dash so the reaction catalyze che thunge ne zo nga zo khal le transferases enzyme la se jo so and next kare hydrolases they are the enzymes which can catalyze the hydrolysis of various bonds water molecule pheche chini ani bond ma breakdown che jo by putting in a water molecule breaking down of bonds for example a glycosidic bond breakdown che jo tinde la jo khalo pension sucrase enzyme do ji by na sucrose la di parallel yo glycosidic bond ma breakdown che yo ila jo khalo yo hydrolysis la yo so sucrase is a hydrolase enzyme ko design dile belong to sucrase la enzyme so so diche in next kare lyases lana they are the enzymes which can cleave various types of bonds by means other than hydrolysis and oxidation mai bache hydrolysis ke mai bache ani thablam shemba ji gi gyu ne khesi bond de ma cha thuye yu ni ba yo bi by na ro and dezu la zu khale gre lyases so there are enzymes which belong to the group lyases right they do not break the bond by hydrolysis or oxidation they break bond by different other means okay diche in the next kare fifth dikhar isomerases lana those enzymes which catalyze interconversion of optical geometric or positional isomers the chemical manda la kho gi sopta chemical arrangement manda shishani isomer manda yorwa so isomer law in khala ore chemical formula chikba deita in sopta this manda che chemical arrangement manda in beking now functional group dige sama pazu right side you get the left side la yu che in bana ko kharcha dure isomer cha dur tinde yo tang thongye jo ma enzyme dige la zu khala ore isomerases so ta thama dikhare ligases lana khuni ni gi dogjo cha da lyases ki dogjo cha dur ligases are the enzymes which can join two molecules by forming covalent bond ligate chawla na jar wala woro so the seu chung so nomenclature dige sama khandes re mangche a segyo mangche ye khare la so enzyme res ro biology na lo la and design ala yawa che da dikhande re there are six large classes of enzymes and they are classified based upon the type of reaction they catalyze seu chung so these are the six different classes: oxidoreductases, transferases, hydrolases, lyases, and isomerases and ligases. So with that, we have finished our discussion on enzymes. Enzyme ka discussion sarda. No. And next, the chapter sawaj go to enda. The biomolecules ka chapter gets sare. You have to go through the textbook exercise questions. Textbook exercise question dige chana tamado. Lo. Oh yeah.